and welcome to the Research Works podcast, brought to you in association with Curtin University and the Healthy Strides Foundation. Your hosts are Dr. Dana Poole and Dr. Ashley Thornton, and together we will interview world-leading researchers in the area of child health to support your practice in being more evidence-based.
Welcome back, everyone, to another episode of Research Works podcast.
Yay! It is actually my favourite time of the week because, I do get to hang out with you, Ash.
Ah, me too, Dana.
It is very cool. And we just get to chat to some pretty cool people about some pretty cool stuff. So that does mean you're really cool.
Oh, thanks, Dana. Yeah. I think you could probably, when my kids grow up, I'll make sure you come around and let them know that.
I definitely will. We've got so much good stuff to talk about. So yeah, I love these. I love these mornings.
Yeah, me too. And we always get to cover some incredible science, and today is no different. We have our special guest, Dr. Tayla Penny, to do that with us today. Hi, Tayla.
Hi, thanks for having me.
Oh, thank you so much for being on the show.
Yes.
I think, we're both, as you can hear, we're both really excited to talk to you about your work, which was actually recognised at the Oz ACPDM PhD platform presentations this year. And, for those of you who maybe aren't familiar with Oz Academy, it's actually an incredibly prestigious platform to be invited to talk at. So the fact that you were invited to this really means that, we all need to be across the work that you're doing. Your PhD covered a lot of different topics, and the main one that we thought we'd focus on today was actually published in Brain Research back in 2020. And that paper is titled Multiple Doses of Umbilical Cord Blood Cells Improve Long-Term Brain Injury in the Neonatal Rat. So to let our listeners know a little bit about Tayla before we get started, Tayla recently completed her PhD in 2021 at Monash University, investigating the long-term effects of umbilical cord blood cell therapies for perinatal brain injury. Tayla is currently a postdoctoral researcher at the Hudson Institute of Medical Research and has some really impressive skills in small animal surgery, behaviour testing, and immunohistochemical analysis. Tayla's work has gained a lot of interest, and the incredible breadth of awards and recognition of late is, I think, a real testament to that.
Yeah, it is actually so impressive when you look at Tayla and the work that she's done, and having seen her on the PhD platform as well is super cool. For many of our listeners, you like us, you're allied health clinicians, we often hear about this kind of science, but we don't often have the opportunity to speak to people like you, Tayla. And I have to say, the small animal surgery definitely got my attention.
I feel like there's probably an icebreaker question in there we could unpack, but I'd already planned one.
Okay.
So Tayla, in case you don't know, we like to ask people a little icebreaker question just to learn a little bit more about you and about each other. And I'll leave the small animal surgery for another day. But the question I thought we could ask each other today is, what is your least favourite chore?
Least favourite chore?
Hands down, it would have to be yard work of any kind. Least favourite.
Oh, that's a good one. I hadn't thought about that one. I've got an extensive list, but yeah.
That's a good one. And I am impressed that you came up with that one so quickly, Tayla. You jumped onto that one. So clearly it's on your mind.
A clear least favourite. I am with you on that.
That's I think I share that one too. But I would say the one that jumps out at me the most, which probably talks about the status of it in my home, is blinds. I do not like cleaning blinds. I can, every now and then you get this particular light that comes to the house and you look up and you look at the blinds and you're like, oh my gosh, shame. Like it is, I can't remember the last time I did it and I just don't want to do it.
Oh, it's so intricate. Like there's so many little bits and there's no
I just want that you can just use just to go. Yeah.
I mean, I've seen a lot on like telemarketing things that claim to be able to do that, but I don't know that they've got one yet.
Yeah. I haven't even gone down the path because you still need to, physically do it to some degree. Yeah. But can you imagine me intricately, like you just can't, like once you get to know me, you know that that's not something I'm about to do, but it needs attention and it's quite bad now. Yeah. What about you, Ash? Because this is a good question.
Oh, look, there's, as you've probably gathered now, an extensive list, but if I had to pick one, it's a real toss-up between folding my washing and putting it away or emptying the dishwasher.
Oh.
I'm fine to like load it and push
get
the buttons
it out of the way,
and get it all done, but actually then like going and putting the stuff away and Yeah, so it probably says a lot about me now that I reflect on that.
I think I've been known to just live out of the washing basket. Like once they've been cleaned, I'm like, why put it away? Obviously, this is my most used, thing. So I just come back to it. And then the same thing goes into the wash and goes back into the basket again. I get it. I get it. Yeah.
I feel like this is quite cathartic, but we'll move on from chores now, I think, and get into the paper. So Dana, could you give us a little summary?
Oh, this feels like a big contrast now from what we just talked about, but here we go, hey? So in this study, the authors examined whether multiple doses of umbilical cord blood could modulate neuroinflammation, decrease cerebral tissue damage, and improve behavioural outcomes when followed up in the long term. So in total, 116 rat pups were included in the study and randomly assigned to the experimental and control groups. So 95 animals were exposed to a hypoxic injury, of which 30 had a hypoxic injury only, 33 had a hypoxic injury and 1 dose of umbilical cord blood, and 32 had 3 doses of umbilical cord blood. 22 animals received sham surgery with no hypoxia exposure. Rats then underwent behavioural testing for motor and cognitive outcomes. Now, the results demonstrate that hypoxic ischemic brain injury in rat pups caused significant behavioural deficits along with neuroinflammation, reduced brain weight, and significant tissue loss. For the first time, this study demonstrated that multiple doses of umbilical cord blood was able to improve long-term neuropathology and induce functional improvements compared to a single dose. And this study provides the first preclinical evidence to support the need for multiple doses of umbilical cord blood for long-term brain development. Whew.
Lots to unpack there.
So good. Yep.
Okay, Tayla, before we start, full disclosure, I've been practicing saying ischemic for the best part of this week because my first question for you is kind of taking a step back from the topic of this paper. Could you just talk us through what a hypoxic ischemic insult is?
Of course. So when we look at a hypoxic ischemic insult in a clinical sense, it is when a baby is deprived of blood and oxygen either during gestation or during the process of birth. So one of the common causes of this is the umbilical cord being constricted during birth. It then blocks off that blood supply and the baby can no longer receive oxygenation to major tissues like the brain. There's other causes that can also occur in utero, but my research more focuses on that birth injury that occurs.
You say it so well because it is it's something that we've often heard of a lot, and the way you've described it there is lovely. So thank you.
Yeah. And when we talk about hypoxic ischemic encephalopathy, or HIE, that accounts for between 8 and 15% of cerebral palsy. Is that correct?
Yeah.
And so there's different phases of the injury that progress over time, isn't there? Could you just talk us through those different phases?
Yeah, of course. So we classify the time at which the injury occurs as the primary phase. And this is when you have the initial blood flow being cut off and a number of mechanisms start to kick in at this point. So you've got cell death, you've got inflammation, and you've also have priming for injury to continue to progress later down this pathway of injury. So that's the primary phase. Then once the blood flow is restored, so this is often the case once the baby's been born, the umbilical cord is cut and then the baby's own circulation starts kicking in. They'll start to breathe and that blood flow is restored. You then have the latent phase of injury. So this is when it looks like the baby starts to recover. A lot of things start to look better. Their blood pressure increases and the baby looks like it's doing better. But then you have the secondary phase of injury, which occurs shortly after that, usually within the first 15 hours.
Wow.
To a couple of days after birth. So this is the most injurious phase where a lot of things are happening, causing injury, particularly to the brain. So you have a lot of inflammation, so both pro and anti-inflammatory mechanisms at play here. You have mitochondrial dysfunction, oxidative stress, all things that are not good for the brain, essentially. So that's our most injurious phase. You then have the tertiary phase. This not as much is known about this phase. It's only been in recent years that people have been really investigating it.
Yep.
But this can last up to years after the initial insult, particularly if the hypoxic insult is really bad at birth. You'll have that prolonged tertiary phase where you have a lot of inflammation and more subtle changes to the microstructure of the brain.
Wow. And I don't think we've heard so much, like you said, you're absolutely right. You sort of hear about that initial phase, which sounds really intensive, but the tertiary phase is something that I don't think I've really, heard anyone talk about before. Why is that?
I think for some time, people were looking at that secondary phase as being like, that's the main injury phase. And once that's over or once we've intervened, then that's it. But the fact that, it continues to progress over time and continues to cause damage is only something that's recently been investigated, mostly through animal studies. A lot of the time people will do a short-term animal study. They'll look at the outcomes and then be like, okay, perfect, that's what it is. But a lot of people don't do long-term follow-up of animal studies to look at those outcomes. And it's also those really subtle changes are hard to see in a human brain when you're looking more on scans and not looking at actual brain neuropathology like you can in animal studies.
Sure.
Yeah. And I suppose, by the time you get to that tertiary phase as well, a lot of the time, the clinical teams working with these children are managing the manifestation of the brain injury, aren't they? They're not.
Yeah.
So I suppose this is a really important piece of that puzzle now that information is out there to consider alongside all of those management issues as well.
Yeah, definitely.
So can you talk us through now about, you've spoken about the inflammation that happens through this process. Now let's just take a shift to umbilical cord blood. First of all, why has it been introduced as a therapeutic option? Like how did that come about?
So umbilical cord blood has been used for quite some time in the clinic, more for like anemia and hematological diseases as an alternative to things like bone marrow or peripheral blood as a treatment.
Right.
But the group that I'm in at the Ritchie Centre, we have been investigating cord blood as a therapy for quite some time before I had even started here. So I kind of jumped in a bit later on this. But the reason we use it is because it's a rich source of stem and progenitor cells that can modulate brain injury and induce repair of the body's own endogenous immune system.
Right.
I think we've probably heard a lot about stem cells, but the idea of linking stem cells back to umbilical cord blood, that's really interesting. So that's the source of those stem cells that we might hear about more commonly. Is that right?
Yeah, definitely.
So to sort of, I guess, to put it in a really simple form, to really understand, the role of umbilical cord blood, it's more like that sort of anti-inflammatory. It's like if you can apply that at that phase, and when you talked about that hypoxic injury phase where there's a lot of inflammation, the role of this now is about this sort of anti-inflammatory treatment.
Right? Yeah. So the umbilical cord blood cells, they don't engraft in the brain. They don't engraft in the tissue at all. What they do is they respond to the injurious environment that you're putting them in.
Sure.
And then they send signals to the body's own immune system saying, hey, you should do this instead. And it causes an endogenous response of the body's cells to help not prevent injury, but repair injury and help with repair.
Right. So it's a very targeted approach. So I guess now I think that the next question sort of has been really just in my mind is obviously we're using this in a rat model. So that's, you sort of touched on that a little bit before, but why it's important to do it in these, these are preclinical trials in rats. But can you go into that a little bit more detail in terms of why we use rats and where then is the status of this information and work in humans?
Yeah, of course. So part of the reason that I use rats is that we can look at the long-term outcomes. Like I mentioned, rats are easy to keep long-term and they're also really good for doing behavioural testing. So we know that in conditions like cerebral palsy, that behavioural outcomes and motor outcomes are important because those are the things that are impaired in these children who have had brain injury at birth. So we really want to understand both the neuropathology of HIE as well as the behavioural outcomes of HIE, and the rat model is fantastic for that. Our group also does a sheep model of our injury, so that's our more kind of clinically relevant model. I don't do sheep work, but the lab group that I am does do that. And so we have looked at umbilical cord blood as a therapy, both in our rat model as well as in our sheep model, to really try and bring together the clinical relevance of our research and make it more translatable into the human population. And then in terms of where we're at in human trials, there have been many trials looking at umbilical cord blood for HI or cerebral palsy. A lot of those are more focused on people with established cerebral palsy, so children, teenagers, adults who have cerebral palsy, and they're giving an intervention later in life.
Right.
But there's been no real clinical trials looking at early life, so just after their baby's been born. But our group has recently started a clinical trial called Cord Safe, and that is looking at umbilical cord blood from the baby, so autologous umbilical cord blood in babies born less than 28 weeks, so they're extremely preterm infants. They're giving the baby's own cord blood back to try and prevent brain injury from developing after an extremely preterm birth.
Wow. Okay, there was a lot of information there, and I think that is super cool though. So in terms of, I guess, looking at umbilical cord blood now, let's get into that in terms of like how do you access it? You know, is it something that, you know, well, maybe we'll go to the rat models. Maybe that's more relevant at the moment in terms of how do you get the umbilical cord blood in the first place? Yeah. Like, how does that work? Yeah.
So for my studies, I use human umbilical cord blood. We have a research collection team who go over to the hospital. We are based at Monash Medical Centre in Clayton. And our research collection team go over, they collect the cord blood from healthy term cesarean sections. So women who are already booked in for a cesarean section they will opt in to donate their tissue, and they'll collect the blood after the cesarean has been completed and the placenta's out. They'll collect the blood and then they bring it back over to me in the lab, and I process the blood to get the stem cells from that.
Wow. Yeah. And how much do you need? How much of the blood do you need to be able to get these cells?
It tends to vary. So not all cord blood has the same number of stem cells in it, but we generally look for anywhere upwards of 50 mils. Around 100 mils is probably my preferred volume to get a good number of stem cells from.
Okay. So that's really interesting. So it's not necessarily, you're not guaranteed to get the amount of stem cells that you might need from any given sample of blood. Is that right?
Yeah, that's right. Yeah.
That's so interesting.
It is so interesting. Okay.
So is there one particular type of cell that you're looking for in these blood samples?
So what we do is we separate out a population of cells called the mononuclear cells. So those are our stem and progenitor cells found within the blood, and they make up just a very small portion of our whole blood sample. And then so within that population, we have a few different beneficial cell types. We have hematopoietic stem cells, endothelial progenitor cells, monocytes, mesenchymal stem cells, things like that, all in that one little population of cells.
Wow. Okay. Gosh.
I feel like the more I learn, the more I realize I really don't know anything.
But it's nice to know the detail, because I think we often we clump it, don't we? When we don't understand the science behind it, we just go, oh, this is just what it is. And, you know, but there's so much detail into it, obviously. And there needs to
intricate,
be. Yeah. Now, is there ever a case where you don't have enough umbilical cord blood to use, you know, in your study or even in a child? Like, you know, because if you're talking about a child that might be really preterm, I imagine you don't have as much umbilical cord blood. Like, is there things that you do around that?
Yeah. So for my current studies, that is not an issue because we're using human umbilical cord blood going into a rat. We generally will combine multiple donors as well. But if you're wanting to take the baby's own cord blood and give it back to them, you run the risk of not having enough stem cells, particularly if we want to give multiple doses, which I've shown is more beneficial than the single dose. So if we want to give multiple doses, babies who are born preterm or growth restricted, they might not have enough cells available for our therapeutic dose.
Right. And is there anything you can do about that?
So that's what I'm currently investigating in my current studies is, it's called stem cell expansion. So taking a small population of stem cells, growing them in a dish to eventually have them proliferate into more cells, increasing the amount of cells we have available to give back to the baby.
Wow.
Oh, that's incredible. Okay, so watch this space, I think is what you're telling us there.
Yeah.
All right. Well, let's move on to the methods that you use for this particular paper. We'll go back and revisit this work we're talking about today. So we've talked about how we collect the umbilical cord blood. Let's talk about the neonatal rat model that you used. So you induced hypoxia in these rats, and then you ended up with 4 groups, didn't you? Could you talk us through the 4 groups that you had?
Yep. So we had our hypoxic ischemic animals, so those that underwent surgery. So we had an injury alone group, so that's showing us what the injury looks like without any intervention.
Sure.
We then had our 2 intervention groups. So one received 1 dose of umbilical cord blood cells. That was given 24 hours after injury. And the other group of the interventions received 3 doses. So we gave those 1 day after injury, 3 days after injury, and 10 days after injury to try and hit those multiple injurious phases that we know occur.
Yeah.
And the last one is called the sham group. So that's essentially a control animal. So they don't have any injury and is a baseline for what normal brain pathology and behavior looks like.
Okay. But in your sham group, they underwent a surgery, didn't they? They just didn't receive the injury to the brain. Is that right?
Yeah, that's right. So we do the surgery, the sham surgery, where we do the same procedures without the actual injury part to control for any factors that the surgery may introduce, like exposure to anesthetic.
Yeah.
You covered all your bases there.
Yeah, that's incredible. My goodness.
And then the rats all underwent behavioural tests, didn't they? So could you explain to us all, because we're, I suppose, familiar with behavioural tests that you might do with humans, but not so much with rats. Could you talk us through what they were?
Yeah.
Sure. So I focus on 3 main behavioural tests in my studies. The first one is called negative geotaxis test, and that's looking at vestibular function and strength and coordination. So for that, we have an angled board. We place the rat with their head facing downwards, and we record the time it takes for them to turn 180 degrees and then walk up the board. So that's what we do when they're quite young, so day 14, so 4 days after injury.
Yep.
The other 2 main ones I do, one is called the cylinder test. So that looks at limb preference, and we use this as an indicator of motor function, which we know is impaired in cerebral palsy. So for that, they go into a clear cylinder. The rats, when they're in the cylinder, will rear up on their hind legs and place their forelimbs on the side of the cylinder, and we record if they are resting on their left or their right limb. So the type of injury that we give, it is an injury to the left side of the brain, which causes weakness on the right side of the body. So the ones that have had the injury are more likely to rest on their left paw and then kind of hold back their right paw.
Yeah.
So that's the one for motor deficits. And then we also do another one called novel object recognition. And this is looking at short-term memory. So we record the rats in a box. They're exposed to 2 identical objects. We then take one away, we give them a new object, and we see if they're going to interact more with the new object, or if they interact equally with both of them, they more than likely don't recognize they've already interacted with the familiar object that's still in there.
Oh, very cool.
Yeah.
I love that.
I've significantly underestimated rats, I think.
Yeah.
And you presented this, which I thought was really interesting. I thought it was worth bringing out. You presented it as an overall behavioural burden using the Z score. Can you just talk us through that a little bit? Because I found that really interesting. I haven't seen that before.
As a part of my PhD, I completed a whole lot of behavioural tests in rats, and then I determined ones that we saw injury, like we saw deficits in based on our injury model. I then transformed all of the tests into a Z score, and that allowed me to combine the Z scores from multiple tests. So essentially what we're getting is an overall indicator of behavioural burden by combining the multiple tests. And by making them Z scores, we're making a standardized score from each test that we can then combine.
That is so cool.
Yeah.
I love that. I think I want to try to do that. I think, no, huge well done on that one. So, okay, so now we come to the all important and exciting results, and we've already touched on it in the intro and a little bit before about the different doses. But can you help us to summarize what the main results were from this particular paper?
Yep. So we, as I said before, we compared the single versus the multiple dose, and we showed that the single dose of cells in the long term was not able to improve any of the outcomes we looked at. So that includes tissue loss, looking at brain immunohistochemistry, and looking at behavior. But the multiple doses was able to improve this. So the multiple doses, it reduced tissue loss of the brain, it reduced inflammation, it reduced cell death, and it also improved our overall behavioural burden for the course of the experiment.
Wow. That's pretty convincing. So when you talk about the multiple doses, obviously that's really highlighting that that's more significant than just a single dose. Now, did you guys have any thoughts about why you thought that was the case?
So I think it's just because we're hitting those multiple injurious phases. So our group has done studies in the past looking at giving a single dose and then doing short-term follow-up. And we showed that in this short-term follow-up that the single dose could improve outcomes. But then when I looked long-term, the single dose could not improve outcomes. So our thought is that the injury is continuing to progress over time. So just giving this one hit of cells is not enough to prevent the injury from progressing. So by giving it at multiple time points, we're able to continue to suppress the injury and promote repair over the course of the experiment.
Wow.
Yeah. And is that really kind of ties back into that tertiary phase of the injury, doesn't it? That, perhaps wasn't acknowledged so much previously, but we appreciate a little bit more the role that plays in more of the long-term injury effects. Is that right?
Yeah, definitely. It's definitely something that I think needs to be thought of when we're designing clinical trials and designing treatments for these kind of injuries.
Yeah. And one of the other things I noticed as well was the impact on kind of specific areas of the brain that you looked at. So, for example, the hippocampus. Could you talk to us about what you found with regards to the hippocampus and why that might be important?
So the reason we looked at the hippocampus is because it is an area of the brain that is often injured from the hypoxic ischemic injury as well as the cortex. And the hippocampus is important due to its role in motor and behavioural outcomes like short-term memory. So we really wanted to see, are we seeing injury there? Can we correlate this to our behavioural outcomes? And are the cells repairing there, which we've shown there is injury there and the cells can repair there.
So I think that's really interesting as well. I think, we talk so much about, and that happens a lot in the area of cerebral palsy in particular, you talk about the motor impairments, and we've started talking a bit more about the sensory motor impairments, and we've evolved there a little bit more. But then in terms of processing and memory, that's starting to evolve. I think neuropsychologists have been jumping up and down saying, hey, got to look at this as well. So it's really interesting that you've picked this out to talk about, the role of the hippocampus and what can happen if there is a hypoxic injury.
Yeah, absolutely. Yeah. The other thing I wanted to touch on, Tayla, is, obviously the doses that you provided to the rats were, initially after injury, and then I think, was it 7 and 10 days?
3 and 10 days.
3 and 10, sorry. Yeah, 3 and 10. And if we were to kind of translate that to humans, because obviously, the life cycle of a rat is somewhat different to that of a human and a child, what would that kind of look like pragmatically for humans?
Yeah, so we would want to give the first dose as soon after injury as we can, or after birth as we can. So probably within the first 24 hours, which can be achieved particularly with allogeneic cord blood, so cord blood from somebody else donated to a baby with brain injury. So probably within the first 24 hours we would want the first dose, because we also know that the earlier we give the treatment, the better the outcomes are going to be. Additional doses, it would probably be maybe one within the next few weeks of life, then perhaps one in the next few months of life. Just really trying to target where when the injury is occurring.
Right. Okay. Well, it's interesting to look at that. Just out of pure curiosity, what is the life cycle of a rat, like by comparison?
Well, so in comparison, my surgery that I do is at postnatal day 10, and that is the roughly equivalent to term brain development for a baby. So day 10 is about birth for a human, but then I go all the way out to day 50, and that's about 18 years of age. So that's roughly the equivalent. Yeah.
Wow. Okay. That's really interesting. Yeah. I'm learning so much, Tayla. So then when you say sort of, providing the cord blood, can I just ask a question about how you actually would provide that? Like how is it actually delivered to the baby?
So for my studies, we just give an intraperitoneal injection, so an injection into the abdomen, and then the cells will travel systemically.
I see.
But for human trials, they give the cells intravenously.
Right. Okay. Yeah.
Yeah. And I mean, obviously as the majority of us being allied health professionals, it's not something that, we should be expected to talk in depth with about families, but these are obviously conversations that kind of come up sometimes, and families are always interested in the different types of therapeutic options that are available to them. So how could we, bearing in mind that it has to be done in quite a sensitive way, what can we say to families who might ask us questions about umbilical cord blood storage from a sibling, and what does this mean for families?
So in terms of banking cord blood, it's something that's quite often done. We work with a company called Cellcare that provide private cord blood banking, and there are also trials that have banked or have used banked sibling cord blood as a therapy for a baby who then is born with brain injury. So I think with the current state of research that we're in, it's not a bad idea to bank your cord blood, but definitely further research and clinical trials need to occur in order for it to become more of a mainstream thing. And that I think is especially true for using the baby's own cord blood. We're not really at a place where we can take cord blood and give it straight back to the baby straight away, apart from the clinical trial we're currently doing that's looking at the feasibility of doing this. But then there is also the potential for, if you're banking your cord blood and your baby is later diagnosed with cerebral palsy, of maybe giving that back, but that doesn't really fall within the early treatment window that we aim for.
Sure. I guess it really does highlight, if we are talking to families as well, that this is in a state of really exciting research and the science that's being developed in it at the moment. But it is, like you said, it's at that preclinical trial where, which is what you've done here in this study, which is why it's so exciting. But now it needs to be really carefully considered in terms of the how and the actual mechanics and the dosage, I suppose, isn't it?
Yeah, definitely.
Yeah. And in liaison with their, their medical team that they work so closely with as well.
Very interesting.
Yeah. So Tayla, where to from here? You're obviously, continuing your work in this space. Could you, and you've talked to us a little bit about some of the exciting things that are happening for you next. Where would you, what direction would you like to see this work go in the future?
That's a good question. So as I mentioned, at the moment, I'm working on this cord blood expansion. So we're trying to truly optimize this for a brain injury setting. The cord blood expansion has been used for other diseases, so more like lymphomas and leukemias and things like that.
Sure.
So we're really hoping to establish this as a treatment for brain injury to then hopefully move more into clinical trials for our expanded cells, things like that.
Wow. Gosh, that's really exciting. And I think there's, watch this space is basically what we're saying. Hey.
Yeah. Lots of exciting things to come.
I think you can see why you got the PhD platform. And so now, I mean, as exciting as a PhD platform is, I would definitely say that maybe the next most exciting thing is probably the Tell It to Ed segment. No pressure. So here we go. We're going to go into the favorite segment that everyone always talks about, Tell It to Ed. Now, as you all may know, Ed is our producer. He doesn't have a background in child health, but he has been listening. He's been recording information and he's been gaining a lot of information information about this area lately, but I think on this one, he looks like he's sweating a little bit. But this will be a great opportunity now for Tayla for you to be able to explain this research in about 60 seconds or so about what you've done. And Ed, you'll have the opportunity for a question. So take it away, Tayla.
For this research, we are looking at umbilical cord blood as a therapy, a stem cell therapy for babies who have developed brain injury and may go on to develop cerebral palsy. So we give the cells and we then hope that they will promote repair within the brain. In my study, I showed that giving many doses of these cells has resulted in improved brain injury in a rat model of hypoxic ischemia.
Well done. It's not an easy thing to summarize. A lot of information. Well done,
Very
Tayla.
succinct.
Very
Wow.
good. No, very good. All right. Well, now the heat is on Ed. Don't worry, Tayla. It's all good.
I'm definitely feeling the pressure on this one. So, usually I think I've got some kind of idea of stuff, but this was too many words with too many syllables in it. I was struggling to keep up. But I do love the idea that obviously science moves things forward, and research, I guess, plays a really big part in that. I've been curious about the whole stem cell thing. I think from a layman's perspective, the stem cells seem like some sort of magic medicine. Like it has the potential to cure so many different things, and yet I still don't know what that is. So I love the idea that obviously the umbilical cord and the blood obviously provides like incredible nutrients and obviously has like amazing properties, which I guess your research is focused on. My question is much more broad. Is Dana and Ash have told me many times about how research takes 15, 17 years to kind of come to fruition. And obviously that's being shortened a little bit because I think researchers and clinicians are a little bit better at translating these things. And thanks to the marvels of the ResearchWorks podcast reaching millions of people. I would like to think that this kind of research and for want of a much better word, technology would be able to translate really quickly. So what happens now from these, pardon my ignorance, rat experiments? I'm sure there's a much better technical term than that. And then to move on to human trials, like how far away am I from seeing this come to fruition?
Oh, good question.
Sorry for the length of it. It's a layman's perspective.
Yeah, it's good.
Yeah. No, that's a great question. So you're right. Research translation into the clinic takes a very long time. We've been working on this kind of research for, as I said before, quite a lot of time. We're very lucky that we work closely with a lot of clinicians at Monash Newborn and Monash Health, and we have some of them as part of our team as clinician researchers. So they have been really integral parts of trying to translate our work from the lab to the clinic. But one of the ways in which our group does that, and not a lot of people do, is we go from our small animal model, our rat model, to our large animal model, which is our sheep model, which makes it then more clinically translatable when we want to move into humans. And that's something we've been able to do. So we've got our cord safe study giving umbilical cord blood to preterm babies. So that's a world first, clinical trial that's happening now. And we're hoping that will then spur on to other trials where we're giving them to babies who have experienced hypoxia in utero and all other types of injuries that we can then give our stem cells to.
So it's close. It sounds close.
Yeah.
Wow.
Yeah.
I love the idea that these things are becoming much more of a reality a lot quicker than they used to in the past. So that's really cool.
Yeah, that's super cool.
And your team sounds like they're taking lots of steps to ensure that it's as translatable as efficiently as possible. So
that's absolutely key. And it's the future direction. It's the current and future direction of how research should be done, shouldn't it? And that's a great example of that.
Yeah.
Well, that has led us to the end of this podcast. I've learned so much today. Like, it was such a good morning. I'm going to be talking about this for quite some time. So Tayla, thank you so very much for joining us and for talking to us and answering our questions. We've really appreciated your time.
Yeah. Thank you so much, Tayla.
That's all right. Thank you for having me. It's been great fun.
Oh, good. Yeah, that's cool. Well, to all of our listeners, remember, if you want to have access to the run sheet notes, you can head to our website, researchworks.net. And there's also a CPD form there that you can fill out if you want to keep this as a record as part of your PD requirements. But for now, we'll say goodbye and we'll talk to you all again next week. See you guys. Bye.
Bye.
Bye.